All right, welcome everybody to this webinar today. This is a continuing series that is put on by the Data Management and Statistics Core of the Arizona ADRC. Just a couple of things to note. This webinar is being recorded, and afterwards the video will be posted to the YouTube page that we maintain for all of our webinars. The link for this video will be sent out as soon as it's posted to all those who registered for this webinar. Anybody who would like to ask questions of Dr. Royal, feel free to post your questions in the chat box, and I will moderate those questions for Dr. Royal at the end of the presentation. Right. So I will go ahead and introduce Dr. Royal. All right, so Dr. Royal is a tenured professor and chief of the Division of Aging and Geriatric Psychiatry at the University of Texas Health Science Center at San Antonio. He graduated from medical school at the University of Texas Health Science Center in Houston and completed training in both internal medicine and psychiatry at the Johns Hopkins Hospital in Baltimore, Maryland. Dr. Royal is primarily interested in the cognitive correlates of functional status and their importance to dementia case finding. He has experience in clinical trials, longitudinal epidemiological studies, autopsy studies, research imaging, and blood-based biomarkers. He has special expertise in psychometric test development, longitudinal studies, structural equation modeling, aging, geriatric depression, vascular dementia, Alzheimer's, and Lewy body disease. All right, and with that, Don, I will turn it over to you, and we are very thankful to have you here and to give this presentation on this really interesting topic today. So thank you very much.
Great, Michael, thanks for inviting me. And unfortunately, none of my talents include IT, so I'm having trouble with my video feed today, but I should be able to share the PowerPoint here. So I don't think that's going to be a problem. Okay, so we'll just get straight to the talk. So I'm going to be showing you how to use Delta homologs to assess dementia-specific cognitive impairment. And the main points here are that Delta provides a continuously distributed omnibus dementia severity metric, which is agnostic to etiology. So I think Delta is dementia's essential feature operating across diagnoses. And you construct it by a unique confirmatory biofactor model and a structural equation model framework. And the latent Delta construct can be output as a D-score, a factor score essentially, and then that can be applied to individuals as a dementia severity metric or used as a predictor or an outcome in different analyses. So just a little bit about Delta's timeline. You know, I'm not a statistician, I'm not a neuropsychologist, I'm not a neurologist. I really came at this from the perspective, I would say, of geriatric medicine. And in geriatrics, there's been a long-standing interest in functional status. And so it was always pretty obvious to me that memory loss was really an inadequate way of understanding the disablement of dementing conditions. And early on, I paid a lot of attention to what we now think of as executive functions. And early in my career, brought out a couple of the first bedside measures of executive function, really shooting to bring executive assessment into routine clinical practice. And the first of these was the exit or the executive interview, which I think to this day is the strongest observed psychometric predictor of IDL in the literature. But it takes about 10 or 15 minutes, and who has time to do that in a routine clinical encounter the way I envisioned it. So we came out a little bit later with clocks, which is a good enough executive test, but has the advantage of being very quick. So that can be deployed in a wide range of clinical settings. But in 2001, I met Ray Palmer, who was recruited to our Department of Family and Community Medicine straight out of Penn State, where structural equation models were first being developed and applied to clinical problems. And Ray upped my game considerably in this area. We've had a very strong collaborative partnership since then, which has resulted in almost 60 peer-reviewed papers over the years. In 2004, we published what's probably the first latent growth curve of change in a cognitive measure predicting simultaneous contemporary change in functional status. But by 2007, I found myself on the horns of a dilemma that I'll explain in a minute. And between 2007 and 2012, Ray and I sort of wandered around in the wilderness trying to explain to our own satisfaction where the variance in functional status is that cannot be easily attributed to cognitive change, at least empirically. And that led eventually to the Delta construct in 2012. So I can't believe it's already been a decade since we came out with our first D-related paper. So here's the dilemma as I understood it circa 2007. So it's easy to show that cognition is associated statistically with functional status, but you have to pay attention to the fact that those associations are statistically weak or moderate at best. So they don't really provide a convincingly satisfactory explanation for changes in functional status. And that put me on the horns of a dilemma. I realized that the glass is half empty, it's not half full. Either cognition does not meaningfully determine, excuse me, functional decline, in which case you'd have to give up the whole idea of dementia. Or there are as yet unmodeled cognitive domains that are responsible for the unexplained disability and most of dementia itself. And that's the path I chose to go down because I could remember from my own career when executive function didn't exist. When I started playing around with that, none of my attendings had ever heard of executive function. There were very few groups in the world that had published anything on that. None of the major neuropsychiatry and neurology textbooks even had a chapter on executive function or even mentioned it in their tables of contents. So, and yet it was right there in clinic, right in front of us, executive impairment, and nobody could see it for what it was. So, so basically in trying to figure out the cognitive correlates of functional status, I chose that route, and that forced me to go back to my basic assumptions about what dementia is. So at its very heart, dementia, I would argue, is not explained by cognitive impairment alone. If it did, then there'd be no such thing as cognitive impairment, no dementia, or mild cognitive impairment. So cognitive impairments alone are insufficient to diagnose dementia. You also have to show that there's disability, and I mean disability in instrumental activities of daily living. And furthermore, you have to show that the two are related. And this leads to a really interesting insight, which is that cognitive impairments that don't explain variance in functional outcomes cannot contribute to the dementia syndrome. They can be the icing on the cake. They can tell you what flavor dementia you have, but they fundamentally do not explain dementia itself. And conversely, that means that dementia is explained uniquely by the cognitive correlates of functional status. And that is what Delta tries to do. Delta is an empirical method of extracting from a battery the cognitive correlates of functional status. So this is a latent variable representing what it means, the cognitive correlates of functional status. It effectively sequesters the variance in a battery of observed measures that is in fact responsible for impairments in functional capacity. And since dementia isn't merely cognitive impairment, but the disability arising from it, then Delta provides an exact empirical estimation of dementia severity that can be used to either diagnose dementia or assess severity, equate different disorders for their dementia severity, or predict future conversions to demented states and discover dementia-specific biomarkers. So it has lots of advantages to look at it this way. So since, I know this is a statistical course, so you all probably know more about structural equation models than I do, but I just want to do a little précis here so that you can keep up with the models I'll show you later. So structural models, as you know, can be presented graphically or verbally in the format of a table, but I have a kind of visual mind, so I like to look at this graphically. And in a structural graphical representation of a structural model, an observed variable, like a memory test here, is represented by a square or a rectangle. So the square variables in these models are observed variables. And we all know, though, that any test, any psychometric test, is vulnerable to assessment bias and measurement error. And so no test measures entirely what it purports to measure. And one strategy to get around that is to redundantly measure a certain domain across several different tests. And between in their aggregate, they're going to get close to finding you what they purport to measure. And one way to represent that would be as a latent variable, which in a structural model is represented by a circle or a oval in this case. So we can imagine memory in the sky, an artificial construct memory function, which is manifesting in this battery of 3 different observed memory tests. But another advantage of structural models is it forces you, the model builder, to account for 100% of the variance in your observed measures. Otherwise, the model won't fit well. And so each test has a residual amount of variance, which is not explained by memory. And in this model here, they're represented as latent error terms. So they're circles as well. So this is a basic measurement model represented in a structural format. We have a battery of tests that are each indicators of a latent measure, which we're going to call memory, comparable to a factor. And it's relatively error-free, not absolutely error-free, but the memory construct is relatively error-free because the error associated with each of its indicators is not shared in common across all 3. So it can't invade the shared variance, which represents the memory construct. So that's memory, but how would you handle something like this? What if you have a battery, a more comprehensive battery, where the tests purport to measure different cognitive functions, in this case, memory, visual, spatial, praxis, and executive function? What would they share, if anything? And there's a name for that in neuropsychology. It's G, or Spearman's G. Or what Charles Spearman, as early as 1904, recognized as general intelligence. That's what he called it. So G represents the variance shared across cognitive domains. And it's been subsequently shown in multiple studies that the G factor is indifferent to its indicators. In other words, G manifests in every observable brain domain or cognitive measure. Spearman himself in his lifetime built G from sensory discrimination tasks alone. No tests of memory or anything, just sensory discrimination across several different sensory modalities, and used the G construct to perfectly predict academic performance in a group of school children. As a matter of fact, there's an entire literature that links G to functional status. Some authors think that functional status is little more than G. And so that literature may be now relevant to neuropsychiatry. Most of it is in child and adolescent psychiatry right now. Spearman invented factor analysis. The guy was a math genius operating at the turn of the last century. He used a slide rule, but we can use computers or even a phone app, which means that you literally have no excuse now not to be deploying latent variables in your routine clinical assessments. And so I'll ask this rhetorical question: where does G fit into your thinking on dementia? Have you ever seen a single presentation at ICAD or CTAD on G and its association with dementia-related biomarkers or treatment effects? And if you're not using this well-validated, really foundational, a seminal neuropsychiatric construct in your clinical thinking on dementia, I'm suggesting that you're flying blind. You're missing something really big about how cognition can be related to functional status and therefore how we can understand dementia. So let's go back to our G model here. The problem is going to be how to integrate IDL into this model. So one thing you might try, an obvious thing might be to regress each of your different cognitive domains onto IDL. And so that's what this would look like. That's what it would look like in a structural model format. So here we have 3 independent cognitive predictors regressed onto IDL, and each of them has been adjusted for their shared variance in G. And I think if you built this model, and Ray and I have built them all by now, you would be surprised to find that there would be weak, if any, significant association between G-adjusted observed measures and IDL. And furthermore, this model would have terrible fit. It's inadequate for thinking about how IDL relates to cognitive performance. So you might revise it now and try this instead. So now you're using the shared variance across a battery of tests to predict IDL. But this model will fit better than the previous one. But what you're saying here is that observed cognitive performance across different domains has no direct path to IDL. The only association between those observed measures and IDL is through G. And that really would change your entire view of what dementia is and how it works. And this model not only fits much better than the previous one, but it doesn't quite fit good enough. It's not a great fit. And then if you adjust it this way, then now you have 2 latent variables, one of which is unable to predict IDL, and the other one, which is D, is a latent variable that represents the shared variance in a comprehensive battery of cognitive predictors that is in fact related to IDL. And now you've sequestered that functionally salient variance in the delta construct. But this calls into question what's left of G. Since we've adjusted all the indicators for delta, G isn't quite the same thing anymore as it used to be. Delta is actually a piece of G. It robs the G construct of some of its variance. And so I indicate that by calling what's left of G, G prime. And so this is the more or less a generic delta bifactor model circa what we've been publishing since 2012. So let's go back and think through this a little bit. We have a battery of cognitive measures. Ideally, they should be as comprehensive as is feasibly possible. We don't want them to redundantly measure a single domain, but ideally they would measure different domains. And then we're focusing on a target indicator. And in this case, the target should be a measure of instrumental activities of daily living. And if you have a set of cognitive predictors sharing variance with a measure of IDL, then you have what I call a D homolog. Now, a homolog is borrowed from genetics. It means a gene descended from an ancestral gene in the same species and that retains the original's function. So, so basically, because G manifests in every observable cognitive measure, there is no theoretical limit to what the cognitive indicators can be in this model. And in fact, empirically, a lot of D homologs have been published from a wide range of cognitive measures. I kept running into reviewers who would say, well, what exactly are the measures you need to build D? And the answer to that is any. It really doesn't matter. It's whatever you have available, whatever is convenient for you to use. And so we had to have a way to equate all of these different unique models, bifactor models, as measuring a single construct. And so we started to call them D homologs. But look how flexible this setup is. What if I just changed the target indicator? So now, instead of predicting IDL, I'm predicting amyloid PET scan results, say in ADNI. So now the latent construct is no longer D. You can't think of it as D anymore. It's not the cognitive correlates of functional status. Now you have the cognitive correlates of a positive PET scan, which could be very interesting and useful. And this would be a paralog, a gene descended from an ancestral gene in the same species that acquires a new function. So if you change the target, you can have some other clinically interesting latent construct, but it wouldn't be D anymore. Now, let's do something else. What if we did this and change the indicators? And now the indicators are imaging biomarkers, different regional amyloid deposition by PET. And what they share with a new target, delta in this case, or D score, now the latent variable would become the imaging correlates of dementia, which would also be clinically very interesting to model, but it wouldn't be D anymore. Now it's something completely different. It's not even the cognitive correlates of anything. Now it's the imaging correlates of a cognitive outcome. And this would be an ortholog, which is a gene independently evolved in a second species that recapitulates the original's function. So in various papers, we talk about D homologs, D orthologs, D paralogs, but that's what we're referring to. It's just shorthand notation to keep track of what is in the bifactor model. So let's go back to D now. In theory, delta can be derived from any cognitive battery that contains a measure of instrumental activities of daily living. Already there, you're in trouble because a whole lot of data sets measure cognition robustly, but they have little, if any, measures of IADL. And you just can't use them to build D. Conversely, you have a lot of data sets in the social sciences that very robustly measure functional status, but they have very limited cognitive assessment. So you can run into problems there. But if you're lucky enough to have a data set that has both robust cognitive testing and functional status, then you can build a D homolog. In practice, even though D is unlimited in what measures you could use, it's been derived in practice already from dozens of cognitive measures, and 17 different delta homologs have been validated to date. And these have been validated by independent investigators in several countries on several continents, involving over 30,000 unique subjects with differing ethnic, educational, linguistic, or cultural backgrounds. Over the aggregate of these studies, delta correlates strongly with CDR sums of boxes, ranging from 0.6 to 0.99 with a mean of 0.83 in 18 different published models. And delta's ROC for the discrimination between Alzheimer's and controls is also very high in every single case, from 0.92 to 1 with a mean of 0.97 in 19 different models. Now, these kind of results are the sorts of things you might see in different artificial intelligence models right now. There's only one truth, and we're getting at it through different means. But unlike AI, D is easy to understand rationally. It's simply the cognitive correlates of functional status, whereas the AI approaches give rise to accurate predictors that essentially are clinically meaningless, and we don't really understand what the model is doing. We just have faith that it's doing the right thing, but it doesn't teach us anything, whereas D homologs can teach you a lot about cognition and dementia. I know you probably can't read this entire slide because the print is too small, but this is just a listing of all the D homologs published to date and the data sets they came from and their validation measures. And I just have 3 or 4 points to make out of this. The first is that it's not just Ray and I. All over the world, people are building D homologs from whatever data sets they have available. These are relatively large databases, so there's not going to be a convenient sample bias. And D homologs have been constructed from a wide range of measures. You're really not limited to what you could put into a D homolog with the caveats that you want it to be relatively comprehensive. In every case so far, you achieve a statistically strong correlation with clinician ratings of dementia severity, operationalized, say, by the CDR sum of boxes, and very high AUCs to distinguish Alzheimer's from controls. And this is across cultures, across languages, across cohorts, across countries, across investigators. Now, let's think a little bit about fit. So another advantage of structural models, in addition to the idea that you have to account for 100% of the variance, is that you can test the fit of alternative models with certain statistical measures. So ideally, we would like the chi-square to be as low as possible, the CFI to be as high as possible, close to 1, and the RMSEA to be as close to 0 as possible. So here are 3 different models, and I want you to appreciate that they're constructed from an identical set of indicators. So these 3 models are different ways of understanding the associations among these 5 observed measures. And the model fit improves as you go from left to right. So the least well-fitted model is a simple multivariate regression, which was the state of the art until about 2007. And so generally you'd see that some of the measures are significantly associated, some of them aren't. Most of them are statistically weak, and only one of them is even moderately strong as a predictor of observed IDL. And in their aggregate, they only explain about 20% of the variance in IDL. And so that was the sort of model that led me to my dilemma circa 2007. So if we arrange this and start to acknowledge the existence of G, then you get a much better fitted model. So basically the data are telling you that you should be thinking about the relationship among these variables as a latent measure that is manifesting in multiple observed cognitive domains. And now G, you can see, has statistically strong loadings. Some of these have strong loadings on G. All of them are significant. And now you're predicting more variance in IDL. But this fit is not perfect. If you turn it into a D homolog instead, now you have near perfect fit. You retain the strong association with IDL, and you're explaining much more variance in functional status than you would with a simple regression model. So some of the D homologs are impractical for clinical deployment. I mean, I really like this one because of its elegance. This was published in 2015 by Alex Kopera in Germany. And in the beginning, none of us knew what measures to put into D, so we tended to just kind of throw in the kitchen sink. We would throw everything in there, and that's what they did in this case. There are 21 different cognitive indicators in these 2 latent variables, and the 2 latent variables differ only by D's association with a functional status measure, unfortunately named the BADL, but it's not a measure of basic ADL, it's a measure of instrumental activities of daily living, and D is statistically strongly associated with it. But that's the only difference between these 2 latent variables, and only D would predict the CDR sum of boxes and have a high AUC for dementia's discrimination, even though G prime is significantly associated with all of the cognitive measures. But because D is indifferent to its indicators, you get to pick them. And that means that you can create homologs that achieve whatever agenda you have, but without sacrificing clinical utility. So here are several that I tend to use in my work. And I have to give them names to keep them straight, basically. So DT2A is a homolog that I can use to replicate biomarker findings between TARC, the Texas Alzheimer's Research and Care Consortium, of which I'm a site PI, and ADNI, which everybody knows is the Alzheimer's Disease Neuroimaging Initiative. TARC was built around ADNI, and so there's a high amount of overlap in their cognitive assessments, and both of these studies assess blood-based protein biomarkers through the same technology, even the same vendor. Although ADNI measures its biomarkers in plasma and TARC measures them in serum. So I find it very convenient to explore biomarker associations with D in TARC and then replicate them in an entirely independent sample in ADNI. And I needed a homolog that would span both of those cohorts. So the DT2A homolog has the same cognitive predictors in both studies and different IDL targets because ADNI, the two studies didn't have identical IDL assessments. But that didn't seem to affect their clinical validation very much, if you look on the right. The DDX homolog, which we'll see a little bit later in this talk, distinguishes orthogonal domain-specific factors. You'll see people develop what they purport as domain-specific indices, but they're using observed measures that are not adjusted for G. So those supposedly domain-specific factors are actually strongly intercorrelated, and the variance they share with each other cannot really be thought of as domain-specific variance. It's actually G. And if you took G out, then you would be able, for the first time, to look at truly domain-specific factors. And what I think you would find is they're weakly associated with IDL, if at all. So I use the DDX homolog to make that point. DEQ is engineered to be factor equivalent across ethnicities. TARC is a ethnically diverse cohort. About a third of its participants are Mexican American. I need to be sure that a D score in a Mexican American is comparable as in its dementia severity to the D score in an Anglo, taking the test possibly in different languages. And so we had to engineer DEQ to achieve factor equivalence across the two ethnicities. Detail can be administered by telephone. I have my psychometricians doing this by telephone in about 20 minutes or so. And I can get a D score remotely. DECOG is derived from items of the test of everyday cognition that Suzanne Farias in Southern California developed. And now you can get a D score and never ask the patient a question at all. You can do it from informant rated items, distinguishing different cognitive factors. And then the D-exit is just, we have the exit score, which I told you is strongly related to IDL, but the exit, the full exit, takes about 15 minutes to do and has to be done in person. And on the exit are 5 items, which could be given over the telephone. So what the D-exit does is build D out of those 5 items, nothing but 5 items from a single set, a test, administered by telephone, and it achieves an ROC as strong as the entire exit score. So that's a nifty little trick. So let's build some D homologs here. Here we have a battery of 8 cognitive measures predicting IDL. And each of the next few slides I'll show you is a slightly better fitting model. So we'll start out with a regression, a pretty straightforward multivariate regression of IDL. It explains about a third of IDL variance. None of the associations are strong. Most of them are significant, but they're weak to moderate. This is, like I said, the state of the art. But now we'll reorganize the very same observed measures as 2 measurement models. One of them purports to measure memory, the other one executive function. Both of those are significantly associated with IDL, but this model doesn't fit well because it ignores the existence of G. You put G in and the fit is strongly improved, but G would rob the other 2 latent measures of their associations with IDL, and yet its fit is still not great. So if you add in D, now you've converted this to the DDX homolog. D is strongly related to IDL. you're explaining considerably more variance in IDL than the initial multivariate regression. And you can see the relative strength of the associations of memory, executive function, and D to IDL. So now we can take these 4 latent variables and test them by ROC for their ability to distinguish demented from non-demented controls. This is TARC data, about 3,000 subjects. And you see we achieve a high AUC for DDX. And in comparison, mutually adjusted memory, executive function, and G prime are much weaker predictors of dementia severity. Executive function is almost completely robbed by the introduction of D to the model. Now, these next 2 slides are a really interesting couple of studies from Brandon Gavitt and his group recently in Colorado, now in Australia. And they fit D to data from the NAC, from the National Alzheimer's Coordinating Center, a data set with close to 19,000 individual dementia workups with a wide range, the full gamut of dementia diagnoses, probable and possible AD, FTD, Lewy body, a bunch of vascular cases, a bunch of MCI cases and normals. And his D homolog is represented on the right. It's indicated again by the FAQ. He has a bunch of different cognitive measures, and he's going to use D to predict all-cause dementia from any diagnosis relative to the non-demented conditions of normal controls and MCI. So he's distinguishing those 2 groups. And D has, the model had really good fit, and D achieves a high ROC for making this discrimination. In contrast, G prime, what's left over in G, is useless for diagnosing dementia. So it's really all on D, the cognitive correlates of functional status. And what's impressive about this is that he achieves such a high ROC across so many different dementia diagnoses. So all of them really. So now compare those results to the results of John et al. A year later from the same group. And they used the same D homolog again, but this time they asked it to distinguish each of the different dementia diagnoses from all of the other ones. So we're trying to distinguish etiology now, vascular from FTD, vascular from Alzheimer's, Lewy body from Alzheimer's. And in every case, D was useless for that discrimination, and it was better made by an index developed from the observed cognitive test scores. So in other words, the domain-specific variance in the observed measures. So D itself, though, was totally unable to make any of these distinctions. And I think what that tells you is that D is dementia's essential feature, regardless of etiology. So the functionally salient variance in a model is represented by the D homolog. And this is going to be very sensitive to dementia severity, but indifferent to the etiology. The information on the etiology is up in the residual domain-specific cognitive functions. So that, Alzheimer's is not the same as FTD. They have 2 different pathologies. They're located in different distributions in the brain. But what they share is the ability to induce functional impairment through cognitive change, D. Where they differ is in the regional effects of their differing pathological burdens, manifesting as changes in 1 or more specific domain-specific cognitive functions. So you can think of the variance as 2 different compartments, diagnostic domain-specific variance and functionally salient variance. But the problem is that domain-specific variance is orthogonal to D, which means it's unrelated to functional status, which means that it does not contribute to dementing severity. So that leads to a very surprising conclusion that disease-specific cognitive variance is not dementing. If you're studying disease-specific biomarkers or the signatures of individual dementing illnesses and the biomarkers of those signatures, you're in the weeds because you're not studying functionally salient cognitive variants. If you were, it wouldn't be disease-specific. It would be dementia-specific, operating across etiologies. So D represents that functionally salient cognitive impairment and is therefore, by definition, dementing. All dementias must be disabling, and so D becomes dementia's essential feature. And that explains why it can't distinguish between any two dementing illnesses. It can distinguish dementia from non-dementing cognitive changes, but it cannot distinguish any two dementing illnesses. And a domain-specific variance is orthogonal to D. So ideally, because you've adjusted it for G, or it wouldn't be domain-specific. Therefore, if it's orthogonal to D, it can't be related to functional status, so it's not dementing. And domain-specific cognitive variance is disease-specific, but all that means is that disease-specific cognitive variance is not dementing. And you'll have to target the cognitive correlates of functional status if you want to find the biomarkers that are functionally salient and therefore make interventions that have impacts on functional outcomes. So let's look at these potential applications. We can use it to assess dementia severity. We can equate severity across conditions or cohorts, even though they might have different cognitive measures in the different cohorts. We can identify domain-specific biomarkers or dementia-specific biomarkers, and we can assess dementia-specific interventions with regard to the functional salience of the treatment effects. If you make memory better, nothing may happen in functional status. But if you make D better, then by definition, functional status will improve. So more than that, D gives you a way to do this empirically. In other words, it's a completely empirical factor score. You don't need a clinician's opinion at all. And that means it can be done now remotely in the absence of clinicians, potentially at scale, across cohorts and etiologies. And because D gives you the full range of continuously distributed dementia severity, then you can do all of this preclinically and think of change in D in its non-demented range as an indicator of cognitive reserve. So you can output the latent variable as a factor score and apply this to individuals. And when you do, you get a nice normal, depending on the cohort you're in, but in this case, normally distributed continuous measure of dementia severity. I like to preserve the standardized scaling because then when you apply the D score to an unknown case, you're effectively wedging them into a reference cohort that's been well characterized and was used to generate the D score measure in the first place. Now you can rank order controls. You don't need clinical diagnoses anymore. You can rank order even normal individuals for their dementia severity. And because it's continuously distributed, you have more statistical power to find biomarker associations. And because D score is completely empirically calculated sum of just weighted observed test scores, you no longer need any expert adjudication. You'll achieve a high association with CDR sums of boxes, regardless of what D homolog you use. And it obviates the need to bring a patient in during a pandemic to a particular visit to be assessed by a clinician with variable expertise who is making subjective opinions about someone's dementia severity and all the complications that implies. This makes it a completely empirical estimation. And even small differences in the D score have clinical salience. So we've shown in this paper from 2017 that each quintile in the D score distribution of controls increases their 5-year risk of dementia conversion by 50%. Whereas each quintile in the D-score distribution of MCI cases increases their conversion risk by a factor of almost 3. So let's look at this distribution in a little more detail. So this is data from TARC. Here are the DEQ scores. Like I said, I like to use DEQ and TARC because it's equivalent across ethnicity. And here on the left is the mean for the normal controls. And now this is the mean D-score for Alzheimer's cases. And here is the mean D-score for stable cases of MCI who do not convert to dementia over the next 5 years. And even within those means are 2 more groups. On the left of the orange is MCI to NC. Those are cases originally adjudicated as having MCI who later converted to normal controls. And their mean D-score, even at baseline while they were diagnosed as MCI, is in between the means for normal controls and stable MCI and statistically discriminable from stable MCI. And on the right of the orange are the cases with initially diagnosed with MCI who later converted to AD. And that group is also statistically discriminable from stable MCI. So the D-score is making clinically meaningful discriminations in the dementia severity of individuals who are already lumped together as MCI, but have different longitudinal outcomes. And so you can also use the ROC to find the optimal point in the D-score distribution to convert individuals from no dementia into dementia. And in the case of this homolog, the ROC was 0.988 for that conversion. And if you were over the threshold, there was a 73-fold increased risk of being labeled by a clinician as a case of AD, relative to being on the other side of that threshold. If we zero in on just the cases adjudicated as MCI and apply that threshold to them, then we find that there's a 16-fold increased risk of 5-year prospective conversion if you're over the threshold into what D calls functionally salient cognitive impairment. But that's another way of saying that D says that these are demented cases already, and the clinicians have misdiagnosed them as MCI. So here are their survival curves. The curve on the bottom is the group that D said was demented at baseline. The other group is the group of MCI cases that D agreed were not demented, and they have very different survival curves. It looks like a quarter of the MCI cases were labeled by D as having a functionally salient cognitive impairment at baseline, not to say dementia, because the clinicians looked at each of these cases and said they weren't demented. But I would just point out that the risk factors for this discrepancy were things that thwart clinical assessments. It was a relatively young age, the absence of an E4 allele, low education, or Mexican-American ethnicity. In other words, maybe taking the test in Spanish or having to conduct the interview through a translator. These are the sorts of things that thwart a clinician's accurate assessment of dementia severity, but D saw through them and was able to recognize a subset of MCI cases who have completely different prospective risks of dementia conversion. So we can also use D for biomarker discovery. And here we have functionally salient biomarkers with enhanced power to detect effects, because we're going to be using a continuous outcome measure. We can look for biomarkers at preclinical stages, even among controls. And we've shown in other studies that the biomarkers you associate with D are replicable across cohorts and biofluids. Usually I use a mediation model approach for this kind of study. So here's a model of the protein, insulin-like growth factor binding protein 2 as a mediator of age's specific effect on prospective D scores a year in the future. So, so TARC has 115 serum proteins. We ran them all through mediation models, just like the one on the left. Because they're prospective, they're arguably causal. This is in an ethnically diverse sample with multiply adjusted associations, Bonferroni corrected for multiple comparisons, and validated through a random split replication. And from that 115 proteins, we can knock it down to 22 that were found to be age-specific mediators or mediators of ages specific effects on dementia severity. And the strongest mediator was IGF-BP2, but the next most strong one was epidermal growth factor receptor. So we've done similar models and published them for several different dementia-specific risks for age, ApoE, GDS, and gender. And in each case, you get a different set of biomarkers, which are empirically mediating the unique effects of each of these risk factors on dementia severity as measured by D. So we get different sets of largely non-overlapping blood-based protein biomarkers. And then in age, for instance, we've replicated them across cohorts in ADNI. So the biomarkers we identified in TARC replicated in ADNI, and that's across a different biomarker, a different biofluid and a different cohort. So here's a nifty way to validate those to show that they generalize. So we're looking now not at D, but at prospective conversion to Alzheimer's from non-demented states in TARC. And we have a sample of 3,500 followed annually for up to 6 years with a mean follow-up almost 5 years. There was a relatively low conversion rate, 9.8%. And we're going to throw all of that into a multivariate regression model of conversion risk, multiply adjusted. And you'll see that age, gender, ethnicity, depression, and ApoE are significant independent predictors of that conversion event, incident prospective conversions. Here are the odds ratios. So in their aggregate, those independent risk factors account for almost a 10-fold increase risk of MCI conversion. We'll go back now to the original model, and what I propose to do is use the short list of mediating proteins that we independently developed relative to D scores and put them into this model one at a time and rationally knock out one predictor at a time based on the specific mediator of its unique effect on dementia severity. And so we'll just skip straight to the final model. And here are those previously identified mediating proteins effectively attenuating the combined effects of age, gender, ApoE, and depression on prospective MCI conversion risk by biomarkers alone now. So, so what I'm saying is that you can use D scores to find functionally salient biomarkers that then can be intervened on to specifically address unique dementia risks. And this cartoon here really summarizes the state of my current understanding of this of the situation. This is actually from a model that I have up and running in ADNI, but I haven't published yet. So here are 10 specific independent D-related risk factors, which in their aggregate explain about 65% of the variance in dementia severity. Several of them, like I showed you above, are mediated through specific serum protein biomarkers. But the main point here is no one of these risk factors is strongly related to D. They're all weakly to moderately strongly related to D, but they're statistically independent. which means that no one is demented by a single overarching dementia process. Any individual's D-score is likely to be the aggregate sum of multiple competing, independent, D-related, and therefore dementing processes. And you're not going to achieve a big effect on dementia severity by intervening on any single risk factor unless you can pre-specify in advance which individuals are demented by the risk factor that you're proposing to intervene on. And I've built up some algorithms to help you do that. And I think I have just enough time to get through them real quick. So let me just run this by you. So what we want to do is select individuals who are demented by the biomarker or risk factor of our choice. So what we have here is a scatter plot of cases, women in TARC. And I had previously determined that one mediator of gender's association with D is serum prostatic acid phosphatase, and that this was a risk factor for dementia only in women. So we're limiting this analysis to women in TARC now. And the scatter plot has on the x-axis detail scores. So this is a D homolog that you could develop or you could administer over the telephone. And on the y-axis are D scores, detail scores adjusted for serum PAP levels. The only difference between these two homologs is that one is adjusted for the biomarker of interest and the other one isn't. If the biomarker had no effect on D, then the two adjusted and unadjusted D scores would be identical and every single case would map to the line of identity, which I've drawn for you. Now, these are highly correlated because PAP isn't driving a lot of D's variance, but it is driving some, which you can see because not all the cases are on the line of identity. The cases who present above the line of identity have been adversely impacted by serum PAP, and now we know them as individuals. We can easily calculate that in this scatter plot, they have a score which is above the line of identity, so they're adversely impacted by the biomarker of interest. And sure enough, they have higher levels of serum PAP. But I identified them through cognitive performance alone. No biomarkers were used to distinguish those two groups. It was just the adjusted D-score and the unadjusted D-score. So once I have that, it's just a weighted sum of observed cognitive performance measures that can be applied over the telephone. So I could screen any woman over the telephone and identify if she is above the line of identity or not, and therefore whether she's likely to have increased levels of this adverse biomarker. And sure enough, they do. But it turns out that a large fraction of women are above the line of identity, almost half of them, and may be adversely impacted by PAP. And not only that, but this adverse effect spans the entire D-score range from normals through MCI and into dementia, suggesting that this biomarker is adversely impacting dementia severity even in non-demented women. And we can now distinguish which fraction of all women those women are, and now recruit them into a clinical trial that would modulate serum PAP and try to improve their conversion risk if they're not demented, or their dementia severity if they are. And you can do this for any biomarker. This is the same analysis applied to a latent inflammation construct that is itself a latent variable indicated by 10 serum proteins, or in the case of ADNI, plasma proteins. And so you can see once again that a large fraction of cases are adversely impacted by inflammation. And now I know who they are as individuals. And sure enough, those two groups, the adversely impacted versus the protected group, differ significantly, not only for their score on the total inflammation construct in both biofluids and in both cohorts, but on all 10 of the individual protein indicators, they're significantly different in the adversely impacted versus the unimpacted group. So once again, we are predicting adverse impacts of biomarkers on dementia severity using cognitive assessment alone, which can be deployed remotely at scale over the telephone, the internet, or any other modality that we wish to construct the homolog in. This is ApoE. It gives you a really nifty looking scatter plot because you can actually see the dose effect of how many ApoE alleles the patient or the subject carries. And the two adversely impacted groups are the groups that carry E4 alleles, and there's a clear dosing effect. At the bottom, you can see the prevalence of each genotype predicted from cognition alone, and that basically the adversely impacted group contains all of the E4 carriers. So that's genetic risk being assessed entirely by cognitive assessment without any genetic testing being required. I'm not even sure if you'd have to disclose that as a genetic risk because you didn't do any genetic testing. All you did is cognitive screening, but you were able to accurately predict which subgroups carry that risk factor. So D has a lot of forward-looking future applications that haven't been achieved yet. We can assess dementia and functionally salient treatment effects for the first time empirically in the absence of clinicians thinking about recruiting a clinical trial under conditions of a pandemic, remotely at scale, and we can even conduct clinical trials against target biomarkers in non-demented persons and start to address their cognitive reserve or their prospective dementia risks. So I think this approach leads to a lot of very interesting changes in the way you can think about dementia. I'm saying that dementia arises from the degradation in functional capacity, not so much cognitive change, and that intelligence, or specifically G, is dementia's essential cognitive correlate. That domain-specific variance, as it can be thought of as being different than G, cannot explain dementia and is never going to explain dementia because it doesn't appreciably alter functional status. And dementia severity, or D score, is not or is seldom impacted primarily by a single cause. In almost every individual, it's going to be overdetermined from multiple risk factors, and you're going to have to deal with that reality in designing your interventions, because you'll either have an intervention that ineffectually improves functional status because it only addresses 1 out of 5 things that are dementing that individual, or you'll have to do chemotherapy. You'll have to intervene on 4 different risk factors independently with 4 different interventions and hope to achieve some aggregate effect. And the only exception will be to identify individuals who are at a high risk of being impacted by the one thing you're prepared to intervene on. And for that, you're going to need a precision medicine approach. So that's that concludes my talk.
All right, thank you very much, Don. We very much appreciate that. We do have one question that came up in the chat. The question is, why do negative correlations exist in the DEQ and the D exit?
The negative correlations. I'm not sure what they mean by that. I guess they mean factor weights?
Possibly, yes.
Yeah. Well, the exit is inversely scored relative to most cognitive measures. It scores on the exit are worse as they get higher. So most test scores are set up the other way. Most, lower performance is adverse. So that would be the most likely explanation.
Excellent, thank you. And we do have, so anybody else who has questions, feel free to put them in the chat, or we do have a relatively manageable group. So if you'd like to unmute yourself and ask your question in person, feel free to do so as well. And Don, I guess while we're waiting, there was one question I had. Uh, so you talked a little bit about reserve, particularly in the preclinical context. And my curiosity was about how one might think about cognitive reserve more generally, in these models that you've proposed, since we tend to think of cognitive reserve being this sort of protective factor for dementia.
Yeah, well, but that's the whole nub of it. You think of cognitive reserve as a protective factor for dementia, and that means for disability. So the only cognitive dimension relative to that issue is D. So other cognitive functions not related to functional status, domain-specific cognitive performance, may have no protective value independently of D. So you could be an MCI case by developing impairment in a domain-specific cognitive task, but that's not the same thing as being a little bit impaired on D. A little bit impairment on D increases your prospective risk of dementia conversion. If you're not a little bit impaired on D, then you're not even in the same ballpark. You see, so it helps you distinguish just any old cognitive change from dementing cognitive changes. And what it draws your attention to is Spearman's G again. So another really interesting implication of that is it would mean that the salient changes in cognitive performance are on a domain that manifests in every observable brain function. That's going to greatly constrain the biomarkers that are of interest, because where exactly could you put a stroke in the brain and simultaneously impact every observable brain function? So it undermines the whole idea of vascular dementia, and it makes you realize why that area has been such a mess. It basically undermines focal brain pathologies and dysfunctions and would emphasize global insults to the brain. So head injury by being hit over the head with a hammer might be a very different kind of proposition from experiencing a blast wave injury in Iraq, where the pressure wave is potentially passing through every single voxel of the brain, and therefore able to impact D.
I see. Thank you.
So it greatly constrains the way you need to think about these things. We have essentially monoaminergic treatments in psychiatry, and cholinesterase inhibitors kind of are working on a monoamine system, but monoamine systems project from particular locations in the brain to particular locations in the brain. And is it, do you think that dopamine or acetylcholine or norepinephrine are positioned in such a way that they can influence the performance on every observable brain function simultaneously? Or are they more likely to affect some tests and not others? Because if they affect some tests and not others, they can't be impinging on G. And therefore, they will never improve functional status, and therefore they can never be dementia-specific interventions, which would explain why cholinesterase inhibitors are pretty lousy as dementia treatments. But instead, we do have some interventions that operate across the entire brain, and those would be mood stabilizers. For example, GABA and glutamate are positioned in a way to impinge upon every observable brain function because they're ubiquitously distributed. And unfortunately, it all goes against us. We have drugs maybe that make dementia worse. And that would explain why benzodiazepines, for example, are such a notoriously evil class of drugs to give to demented persons, because maybe they adversely impact your D score. I mean, I'm just guessing because I don't have any data sets that allow me to look at that yet. But that's what I would guess, is that alcohol and and benzodiazepines adversely impact G and D. And that's why, if you think about intoxication, you're looking at something that impinges upon multiple clinical functions. It isn't just memory that's impaired when you're intoxicated. It's memory and executive function and language and judgment and balance and sensory performance and emotional processing. It's gait. You see what I'm saying? It's every observable brain function.
Mhm.
And so that's much more likely to be a model for what the fundamental dementing process is like.
Thank you. Any other questions for Dr. Royal? All right. Well, thank you, Don. I really appreciate you taking the time to give this talk today. It was very informative and very informational. The talk itself is going to be posted on a YouTube page that I maintain for the Arizona Alzheimer's Disease Research Consortium, particularly for our data management and statistics core. For everyone who registered for the webinar today, I will send out the link once the video is uploaded for it. All right, and thank you again, Don. Thank you again, everybody, for your attendance. Just to let everyone know, we do these webinars quarterly, and we'll actually be taking a break in July so as not to supersede with the Alzheimer's Association International Conference, but we will start up again in October with a very good talk that I'm hoping to have lined up for then. So thank you all very much and have a good rest of the day.
Okay.